Good morning. Can you hear me? Is the micing good? Okay. So today I'll be talking about DNA identification, the search for truth, and what I'd like to begin in this talk by doing is explain what the title means, because I know not all of you are forensic scientists with probability backgrounds, right? Safe bet. So first, DNA. What's DNA? So here's a quick cartoon to lay that out for you. So as you all learned in high school, there are cells, and cells have nuclei. And inside the nucleus, we have chromosomes, 23 pairs of chromosomes. And if you take a look at one chromosome and blow it up, on that chromosome, there are many genes or genetic locations where there are inheritable traits. Some of those, let's take a look at one locus, one of those loci, come over here. And since DNA comes in pairs, one from the mother, one from the father, and so on, the value of what the DNA content is at any genetic locus is called an allele. So we have two alleles. Here they are. And this particular type of genetic marker is called a short tandem repeat, or STR, which is what's prevalent in DNA identification. Typically, scientists use 10 to 15 of these as junk DNA. The reason they're junk DNA is what we're interested in is random repeats. And the way this works, it's like an accordion effect. You get 2, 3, typically 4 for forensics of units of DNA that just repeat 8, 10, 12, 15 times with a lot of variability. The reason that's good for identification is if you have maybe 10 different lengths, say 10 through 20, and you have 2 alleles in the allele pair, then there are maybe about 100 different possibilities. So if there are about 100 possibilities, there's a certain relative uniqueness to somebody having one of those. And if you multiply that by 10 or 13 markers, that's where you get numbers like a quadrillion to 1. Okay? And so in this case, if the length of one marker is 8, the length of the other marker, the number of repeats, is 7, then the genotype, which we'll be referring to throughout the talk, is a 7 and an 8. Okay, so the genotype of this cell would be a 7 and an 8. Okay, so how do we turn the genetic markers that are present inside a human cell into something that is a genotype that we can observe? There are 3 levels. First, there's the level of reality. Here we have biological evidence. In the real world, we believe as scientists that there really is DNA present there, and we're going to see what it is. To see it, we have to collect data, so the laboratory transforms for us that DNA, say from the blood evidence at every locus, into signals. This is a cartoon of what a signal will look like. At the end of the talk, you'll see some real DNA signals. And if there was a fragment of 10 repeats and another allele of 12 repeats, then you would see in a length assay on a DNA sequencer that one length would go out to the number of base pairs corresponding to 10 repeats, the other one out to 12 repeats. So we can measure peaks or heights corresponding to the quantity of DNA on a scale that is the size of how long the DNA, how many DNA letters there are. Then, this is a very easy situation where it looks pretty unique. We can infer from the question data what the question genotype would be. We'll call that Q for questioned. And in a simple case with pristine DNA, we get a unique genotype. Only after you've inferred the genotype, never having looked at a suspect, because that would bias your results and invalidate your answer, can a comparison then be made to a suspect genotype, and then you can determine to what extent they match. So if that was all DNA was, it wouldn't be very interesting, and the problem would be solved. But most DNA evidence doesn't look like that. Most DNA evidence comes from mixtures. Most labs, for example, sexual assault is the most common crime that people have been looking at, and that's inherently a mixture of at least 2 people, say a victim and some other perpetrators. There can be 3 or 4 of these people contributing. There's also low-level DNA, where the amount is low with a lot of random variation. And there can be burnt, damaged, like in the World Trade Center project DNA. So let's just stick with mixtures for now. The point is there's considerable uncertainty in the DNA itself as to what might be there, and that translates from the lab into uncertainty in the question's data. We no longer see two clear peaks, one or two clear peaks, telling us what the one or two alleles are of the one individual. There isn't one individual. And so depending on how that would be interpreted, and I'll go into that a little bit later on, but not in great mathematical detail, we would infer from that data, along with another 10 or 15 experiments of signal data that showed the size of the DNA length, the alleles, along with the heights and how much, using the quantitative data, what the genotype is. Well, this is our inferred state of the real world, and when we infer and there's uncertainty for the last few hundred years, scientists use probability. So we can't say we know for certainty what it is, but of the hundreds of possibilities, we might be able to list three, say a length of 10, a length of 12, an 11, 12, a 12, 12, and by hand or better with computers, assign them probabilities. That probabilities are weights or values that are given to possible events or hypotheses that must add up to 1. And so they would have different values. Then we could take that genotype Q of the questioned evidence and then make a comparison with a suspect, or as we'll see later on, maybe with many suspects, and determine the extent of match. Okay, again, this has to be done objectively. You start with material, you generate data, the inference is done without ever seeing any suspect. Actually, a good analogy for this crowd would be, it's kind of like if you're looking at an atom and you're looking at its quantum state, right? It's an electron cloud. So you have a bunch of probability levels of what the energy states could be, some distribution of where they might be. And until a comparison is actually made with something else, with a photon passing through or with another atom, there is no match. There is no comparison made. There's only a probability distribution that has the potential for comparison. Again, I'm just walking through the title, so now I've explained what DNA is and identification. Now, let me talk about what science is. Why not? So what does science mean to people who've written books about what science means? William Jevons wrote a book in the Victorian era, 1874, that was a classic that was very widely read called The Principles of Science. And about 100 years later, a famous physicist, Richard Feynman, who many of you would know about, wrote a book called The Character of Physical Law. And in their section about how do you infer knowledge about hypotheses of the world, interestingly, they say exactly the same three steps and basically the same words. First, you propose a hypothesis. The hypothesis can be based on any theory, no matter how incomprehensible. It's just some hypothesis. What's most important is the predictions that the hypothesis makes. You examine the consequence of that hypothesis, and then you compare the consequences, what's computed from those assumptions, that hypothesis, with data. And if you get really good agreement, then there is more support for your hypothesis. It becomes more likely. If it doesn't agree at all, no matter how much you love your hypothesis, well, it's just unlikely, and it might be rubbish unless you made a mistake. And in between, sort of agree, we get shades of gray. You know, maybe it's there, maybe it's not. And this is how science works. Now, what's interesting is what happens when you're not looking at just a pair of hypotheses, is one more likely than another, but you're looking at all possible hypotheses, which we can do in forensics, particularly in DNA. And here, the search for truth was probably initiated with the Reverend Thomas Bayes in 1765. So Bayes solved a philosophical problem mathematically that was published posthumously, in which his question, in science and in logic and dating back to the Greeks, the concept that we have some hypotheses and some assumptions, and from there we can infer what our conclusions are. If it's probability, if we assume that a patient has a fever, then we can make some assumptions about what that fever might be, or going backwards. If we assume that someone has an infection, then they have a fever of a certain type, and there's a mean and a standard deviation as to what the values would be. That's reasoning from hypotheses to observations. That's what most of math and logic is set up for. What Bayes asked was, how do you reason backwards? How do you figure out the causes? How do you look at the causes by examining the data? Maybe the patient has an infection. Maybe they have an inflammation. Maybe they took some pills. Who knows? How do you find out the causes from the data? And that had not been worked out rigorously with probability until 1765. And this is Bayes' theorem. This isn't a principle or a heuristic or a guideline. This is, if you accept the basic axioms of probability, this is a fact of life. This is a theorem that comes from the heart of probability. It says we have some belief in a hypothesis, maybe the genotype from the data, or whether it will rain tomorrow, whatever the hypothesis is. And then we examine data. And the data updates our belief to a new state of belief that's been informed by the data that we've measured, observed, heard about, or whatever. So a way of stating Bayes' theorem is that our belief in a hypothesis after we've seen data is proportional to, as in multiplying and proportional, to how well the hypothesis explains the data, that's called the likelihood, for those of you who know that, times our initial belief, called the prior. And so we update our beliefs and hypotheses by examining data. Now, to make this work, you must consider all possible hypotheses, not just the ones you like. And in order to do that, you need computers. When these methods were first invented during World War II with the first computers and statistical reasoning, and actually made practical, because you need computers to do this, the first problems had maybe 10 or 20 variables. Biology problems, including forensic DNA, maybe have hundreds or thousands of variables. Physicists work on problems with millions of variables, and they all interact in complex networks. But the computers can now solve this and find the probability of the causes by examining data. So whether or not you know it, you've just been spared a lecture on Markov chains and probabilistic modeling and hierarchical. If you like that stuff, great. Okay, so how do we measure the information that we gained when we end up comparing an inferred genotype to a suspect's genotype? We use a form of Bayes' theorem that measures the information gain. It's often called the likelihood ratio, or LR, for reasons I won't go into. The hypothesis that we start with, that we're trying to examine whether we're doing a police investigation or testifying in court, is the hypothesis that this particular suspect contributed their DNA to this particular evidence. That's the hypothesis. And we always compare with another hypothesis. In this case, it's the alternative, that someone else contributed their DNA. And the information gain, or the likelihood ratio, can be described as the odds after we've seen the data divided by the odds before we saw the data of that hypothesis. In other words, before, we may have had some belief. We may have been the police and believed he was guilty. We may have been his mother and believed they were innocent. We may have been on a jury and believed that we're impartial. Then we examine data, and after the data, we have a belief afterwards in what the hypothesis is. This is just like energy states, right? You may never know what the absolute states of energy are, but you know what the differences are. And the likelihood ratio measures the difference objectively, speaking only to the data and factoring away any prior prejudices that you might have, which is why it's so powerful in science and particularly in the courts. If you take the logarithm of the likelihood ratio, then numbers like million become 6, 10 to the 6th, and it turns out to be nicely additive, and information scientists and statisticians and many forensic scientists virtually only work with the logarithm of the likelihood ratio, which is additive. Of course, saying that the evidence against the suspect is 12 is less dramatic than saying a trillion, 10 to the 12th, but scientifically it works to use those units. So you can think of it as the order of magnitude of the power of 10. So let's talk about DNA mixture data. Imagine, as I showed you, that we have different sizes. Maybe you have alleles of 10 repeats, 11, 12, 13, some number of repeats. And when you assay them, on a DNA sequence, fluorescent DNA sequencer, you see the different amounts. But before we even look at the data, let's just draw a cartoon of what's actually there. Suppose contributor A actually has this larger amount of the two blue alleles, and contributor B has a smaller amount of these two orange alleles. And that's what's actually in the tube before you ever do any experiment at all. Then, when you amplify them with PCR in the laboratory, you would get data that looks like some combination with some random variation of the larger amount of the blue contributor and the smaller amount of the orange contributor. You would get data that reflect what's actually going on. This is mixture data of the blue genotype and the orange genotype mixed together from two individuals. Okay? Now we want to interpret that, and that's where life begins. So the lab part's very straightforward. You just generate data. They do a lot of work. Different countries do it differently. Some countries like to generate data in duplicate or triplicate when there's a question. The U.S. generally doesn't. So there are issues with data, but for the most part, once you have the data, the inference problem is what are the genotypes based on the data. So what I'll first talk about is quantitative DNA mixture interpretation. You don't read about this in the paper every day, but in forensics, it's been a hot topic for 10 years, and it's been heating up even more as the amount of DNA is getting less and less with property crime and other sorts of evidence that comes in where there's tremendous uncertainty and variation in the data. So we want to infer a genotype. And to do it, using Bayes' rule, we have to consider every possible allele pair. That's required. If you don't consider everything, then the rule doesn't work. And then we compare what's predicted, the consequences, the pattern that comes out of assuming a certain genotype with the data. That's science. And then the rule is a better fit's more likely it. That's just the likelihood principle of statistical inference. So if you recall, this is what the data should look like if everything were perfect. Then if that's what we observe, when we make a prediction, so it matches the data, it has a high likelihood. If we start moving things around and say, well, maybe we agree that the purple individual, the blue individual, has that much DNA at those two alleles, but the other individual maybe has something there, something at allele 10, something at allele 12, is missing. That doesn't really fit the pattern as well, so it would have a lower likelihood. And if you do this quantitatively and you generate all possible patterns by considering what the allele pairs are, the genotypes, all candidate values, and you consider all possible quantities of how much would be there. And if you're a forensic scientist, how many people here are familiar with forensic DNA? So you would know that there are second-order effects like PCR stutter and peak imbalance and degradation, all of which are considered by a computer but not discussed in this talk. Then you can list out what all the possible allele pairs are. Some will have a higher probability, some will have lower, most will have pretty much close to zero because they have no support in the evidence. And you get a probability distribution. And you need computers to do this, to consider all the main variables, the secondary variables, and with modern statistics you can also consider the uncertainty of every variable itself. That began happening about 20 years ago with computational statistics, where you're able to consider the uncertainty to just be one more parameter. And that's when things got very interesting. Okay, so what's the information gain? Well, we make a comparison with the suspect's genotype. If we're comparing with a hypothesis that it's the suspect who contributed, then we don't care about anybody else's genotype. We only care about the suspect's. That's part of the likelihood ratio concept. And we ask, what was the information gain or the likelihood ratio at that one of the 15 genetic loci? There are many ways to compute a likelihood ratio. One of them is to look at the probability of that genotype value, the allele pair, after you saw the data than before. These things all sort of look the same after a while. You know, it's conditioned on the data. And so before I ever saw the data, if I just saw an individual, what might I believe? Well, I might believe that it's whatever the population value would be. Like if that allele pair has a prevalence of 5% in the population, before I've examined that person or some evidence, then my belief would be it's 5%. But if after I've seen the data, I now believe that it's gone up to 50%, that ratio of 50% to 5%, which is 10, is the likelihood ratio. It's the information gain at that locus. And the logarithm of 10 base 10 is 1, so it would get 1 log unit of information. Now, computers can do this very objectively. They infer the evidence they infer the genotype from the data without ever looking at suspects. And then when you make the comparison with the suspect to compute a match score or likelihood ratio, that's automatic. You just plug it into a formula. It's determined by the suspects you're comparing with. So there's a study that we have that was recently accepted. It was done in collaboration with New York State. And what we did is we looked at several groups of cases and compared what the state of New York had recorded in their case folder relative to what the computer did. It's a very clean study in that we are comparing against adjudicated cases with fixed historical results. So suppose we look at these cases of 8 mixtures and we analyze them the way New York did, assuming that we don't know anything about the contributors. So we're solving for 2 unknown genotypes, each genotype corresponding at all the loci corresponding to the contributors. How much information did we get? Well, here are the 8 cases, and here is the information, the log of the likelihood ratio. So 10 is 10 billion, and 15 is quintillion. They all have names, but certainly in science, looking at 5, 10, 15 is a little bit easier. And what we see is a certain amount of information, and on average, we have, what's this, 10 to the 13th, which is about 10 trillion. So that's the efficacy. Now there's a qualitative review that people do. And you may find it hard to believe, but what people really do is they take all the quantitative data, peak heights at 120, peak heights at 2,000, peak heights at 50, and they take a line and they draw a line through the whole thing that's set by looking at a whole bunch of unrelated cases in their lab. And then everything that's under the line doesn't exist, and everything over the line is assumed to have the same height. It's not the world's most informative procedure, as you're about to see. It's an exercise in how you discard data. The higher your threshold, the more data you throw out, and of course all the quantifications thrown out. People are looking at me in disbelief. Fred, could you say that people really do this? Yeah, this is prevalent practice in the world. Certainly in the U.S., it's the only method that's done. People don't look at quantitative data. And so in this situation, when they make an inference, they would try out all possibilities. If you see 4 peaks in your data and you take all possible pairings, there are 10 of them, you just try them all out, and with this method you say they're equally likely. The rule is every allele pair has an equal share. And therefore, everything has an equal likelihood, and you're not getting any information gain at what the data is trying to tell you is the correct answer. You're just saying everything's the same. And the result is you lose information, and the lower genotype probabilities, as we showed, that the information gain is related to how much increased belief you have in the genotype when you're done. If you're dispersing your belief among lots of impossible possibilities, then you end up with a lower information gain. So that's what we see. In orange, you're seeing on the same cases what New York State reported. And you should know New York State is one of the top labs in the country. They solve cases more aggressively with very tight controls, and certainly are looking at all their data. We don't see them ever making mistakes. If they have any question, they won't call it. They call as much as they can, but no more. So assuming 2 unknowns and solving for 2 genotypes, if you look at the heights and what the difference is here, so this is about 1 million, that's about, I don't know, quadrillion, and you look at the differences, the average difference in log units is over 6. So there's about a loss of information of over 1 million. That's what's generally seen with these threshold-based human review methods. If you don't look at the data, if you make everything black and white, you start losing. Okay, and how reproducible is the computer? I'll go into how reproducible people are at the end of the talk. The computer is reproducible within a few tenths of a log unit. This is a within-group standard deviation across the data that's computed. So how much variation do you get on the same case? So in blue is one run on the computer, is green is another run. Why is there any difference at all? Because we use statistical sampling methods, so we don't expect two answers to ever be identical, but we do expect the information from them to be close. And as you can see, they are quite close. Okay, what about if you have a case where you know the victim's genotype, for example, in a sexual assault or a homicide, where you have the victim and you can reduce the dimensionality of your problem by putting in, I assume, the genotype of one person. Now I'm looking for one unknown genotype, not two unknown genotypes. There's a gain in information overall. It goes up to about 10 to the 17th. And when you compare with people, people also get a gain in information from about 10 to the 6th to about 10 to the 12th. So when the victim profile is available, it's generally informative to use it. That said, many labs have protocols that preclude them from ever using a victim profile, not New York State. So here, the difference is no longer 10 to the 6th. It's 10 to the 4.5, which is about 50,000. Okay. Another way of looking at this is that when you infer from data, you can never add to what's there if you're doing it objectively. You don't know what the answer is supposed to be. But if your inferences do things that are less efficient with the data, you can always lose information. So we know that the computer is doing some level, and the question is, if that's a gold standard, how much information are people losing when they do human review in daily practice in crime labs? Again, it's reproducible because there are fewer variables. The reproducibility goes down, decreases to the level of hundredths instead of being at tenths. So it's a highly reproducible method, very informative. And here's a summary from this validation study. So what did we see? Quantitative computer versus qualitative human review, either with two unknowns without the victim or one unknown with the victim. The computer averaged 10 trillion with a small standard deviation within group. People averaged about 10 million. So the information gain was about 6 log units or 1 million to 1. And when it was a simpler problem and the victim was known, looking for only one of the two contributor genotypes, then there was an increase in information in all methods, but the average information increased 4 log units with computer review and increased 5 log units with human review for a gain of 50,000. So that's the summary of the validation study. Okay. As far as we know, it's the first time anyone's bothered to do a detailed comparison of quantitative methods where we use or try to use all the quantitative peak data versus the essentially 100% prevalent crime lab methods of discarding it and just going with all or none events. It's worth talking about how this plays out in court. For the last few years, I've had the fortune or misfortune to have to testify about this stuff in court. And it's fortunate that you have an opportunity to explain to non-scientists, juries, lawyers, and so on, what is going on, what the science is. That's fortunate. On the other hand, never fun. So here's a case, Commonwealth of Pennsylvania versus this trooper, Kevin Foley. So this is the Blairsville's dentist case where the dentist was slashed in his home and bled out. Cyril Weck did the autopsy, and the main suspect was a state trooper. And Kevin Foley, it's Kevin, actually doesn't look at all like the Reverend Bays, was charged with the crime. And the FBI was sent the data because there was a possible conflict given that he was based in Greensburg where the DNA crime lab is. And the FBI came back with a statistic of 13,000. There are about 13 million people in the state of Pennsylvania. And so if the DNA was the only evidence in this case, or the primary evidence, there was a sort of a problem in making any convincing argument from the data. What were the data? Well, the DNA evidence came from under the victim's fingernails. They were, you'll see the data in a second, or some of it. There were clearly two contributors to this DNA mixture. Overwhelmingly was the victim's own DNA that comprised over 93% of the evidence as it was calculated by the machine. And there was an unknown 6.7%. You should be aware that most crime labs are very uncomfortable calling any genotype that would belong to a contributor that's less than 50%. Dropping down to next to 5% was just not any normal interpretation practice in the US at this point. There was a good amount of DNA, 1 nanogram or 1,000 picograms in a standard volume. The analysis was done with standard kits, Profiler Plus and Cofiler. We know the victim contributor genotype because we have the victim. And so the question is, can we solve for all the variables in the case, all in the data, the uncertainties and the mixture weights and the quantities and the stutters, but most importantly, the variable of interest is the genetic identity. It's the genotype, the probability distribution of allele pairs at every locus of the unknown contributor. So we ran it through TrueAllele, and it didn't take a few minutes. It took like 6 to 10 hours. It was a harder statistical problem. And it assumed that genotypes add. The way to think of it is that in science, we often believe that things are proportional. 1 + 1 = 2. And so that's the novelty here, is that as opposed to what current practice is. We're adding up the amounts of DNA. After we've inferred the unknown genotype, some comparison is made with the suspect genotype. You may say, well, isn't that obvious? There are some recent papers in the Journal of Forensic Sciences that indicate that most human review of DNA data is looking at the evidence and comparing it with the suspect in order to infer what's there. And the papers don't commend this practice. They describe what the potential statistical and inferential problems would be with such practice. There were 3 DNA match statistics. The inclusion statistic, which is just, are these pairs of alleles included in what we're seeing in the data? And from Bayes' theorem, you already know there's danger with just trying to infer directly from the data without using a little bit of mathematics like Bayes' theorem. I'll show you how that worked. It was 13,000. Dr. Robin Cotton, former director of the Cellmark Boutique Laboratory in Washington, looked at it, and she made some other assumptions, and that number was 23 million. I'll go into that in a bit. And then using linearity addition, the concept that the amounts of DNA add up to help explain what the quantitative signals are that we're seeing, gave a score from the TrueAllele computer of 189 billion. So what I'll endeavor to explain in the next 10 minutes is why do we have different match statistics? How do the different mixture interpretation methods differ? And for those who are interested, what would you say in court so as to not confuse people? This is how the methods differ. Of all the data, we can break it down into 2 categories. Are you using the victim genotype or not? And are you looking at the quantitative data, or are you making all or none decisions, slicing off the data and saying, I think that something's there and something's not there? The inclusion method does not use the victim profile and does not use quantitative data. Therefore, we'd expect it to be the least informative. The subtraction method that Dr. Cotton used does use the victim profile, reducing the problem to searching for one genotype, but again, uses thresholded data. And the computer methods, as scientists, we try to use all the data that we possibly can. In court, there's an admissibility hearing asking to what extent is what you're showing to be reliable science. There are 2 standards. The Daubert standard is based more on validation studies and as a method testable, has an error rate been produced, is it peer-reviewed. In Pennsylvania, the method is more on general acceptance, the Frye standard, which deals with to what extent are the underlying principles accepted in the relevant community. Here, the court determined the relevant community was the forensic statisticians and mathematicians who create the methods of DNA interpretation, not the practitioner community. And we went through, looking at the literature, we gave them a CD with over 60 articles on it, different PDFs, and explained to the judge, this precedes the jury, that quantitative STR peak information has been published ever since STRs existed about 30 years ago. Genotype probability distributions date back to the beginning of genetics. I don't have a picture of Mendel, but if you think of the P's, you didn't say, I know that that P pair is going to be green or shriveled or whatever, or I refuse to say what it is because I don't have absolute certainty. Instead, you draw a Punnett square and you give answers like 25%, 50%, and you're quite happy to do that because probability is the language of uncertainty in science. There's been computer interpretation of STR data since the 1990s. Statistical and modeling and computation has a great history with lots of stories and people who've done things, which I don't have time to go into, but I don't know if the judge loved the stories, but I told them anyway. There's a nice literature on the likelihood ratio, which in its modern guise came out of cracking the German Enigma code by Alan Turing in the 1940s for the British. The mixture interpretation admissibility, there had been a court ruling that said you should always consider interpretations but challenge them in court with cross-examination. There are a number of systems that do quantitative DNA mixture deconvolution and other groups that similarly do probability modeling and stochastic search with computers, had recently published papers. Those were very helpful. And of course, we have our own publications. So the evidence was admitted into court. And the main defense argument in this case was that how could you possibly have a score in thousands, a score in millions, a score in billions? Clearly, this didn't make any sense. And what you expected was that the other methods should just give you a score measured in tens of thousands. That's what you would expect. Now, of course, we argue that if you use more, and so does the whole literature on this in forensic science, that if you use more of the data, you would expect more. But fortunately, we had done a study, which was published in December of 2009. I'll show you another slide from this soon. But let me explain the slide. This is, whenever you get a straight line out of data, it's always worth talking about because it's so unexpected, as some of you might have figured out with your projects. So let me tell you what you're looking at. Here's the amount. First, what are these points? We had DNA samples that were made up for us by the National Institute of Standards and Technology, NIST, as part of a National Institute of Justice, NIJ, funded study that Cybergenetics did with a number of collaborators. What NIST did is they took two pairs of known individuals. They created mixtures in proportions of 90-10, 70-30, 50-50, 30-70, 10-90, and then then we had protocols where we diluted the 1 nanogram of DNA to, we had 1 nanogram, half, quarter, and an eighth. So we had 40 mixed diluted samples of known composition that we could interpret by a computer without ever looking at what the answers were, and then compare them afterwards. And each of these blue dots represents one of those 40 interpretations with two measurements on it. So the first measurement is how much DNA was there. That's the x-axis. And in particular, we're looking at the amount of culprit DNA. How do we know what it is? Well, we know what the mixture weight is, and we also know how much DNA there is. So we know if you have, for example, a 50-50 mixture and you had 500 picograms of DNA, then half of 500 would be 250 picograms. So we know where every point is along the x-axis. And then we looked at the likelihood ratio, the measurement information, when the objectively inferred genotype probability distribution was compared against a suspect. And this, again, here's 1,000, million, billion, trillion, and so on. We plotted them on log scales. So here we have 10, 100, 1,000 on a log scale of mass of DNA quantity, measured in picograms. And here we have the usual log information scale of the likelihood ratio. And this is what we saw. For the cases that had more than 100 picograms, in the situation, they're analyzed 4 different ways. This way was solving, assuming the victim and solving for the one unknown using quantitative interpretation. I'll show you later what qualitative human review does. But with quantitative review, down to 100 picograms, we're basically recovering the full 10 to the, 20th, 10 to the 18th information that's present. And then an interesting thing happens. As we go down below 100 on a log-log scale, we have a straight line regression that tells us how much information we're losing as the amount of DNA decreases. This was a very new result because, as you can imagine, people don't typically quantify what they're doing very much in terms of the information or the interpretation. And if you take 1,000,000 to 1, see, as the likelihood ratio, that's the convincing threshold, which legal scholars report that juries beyond 1,000,000 to 1, it doesn't make much of a difference because nobody can really think about much more than 1,000,000. Then where that curve intersects 1,000,000 to 1 is a measure of the sensitivity. And so what it shows is that quantitative interpretation with computers in this situation, solving for one unknown, goes down to about 15 picograms of DNA. Okay? That's neither good nor bad, but human review guidelines are uncomfortable with anybody going much below 100 picograms. And all that evidence is generally thrown out or not interpreted by crime labs. So we could analyze these quantitative results and then do something else. If the FBI had quantitated that 1,000 picograms of DNA, and we had determined that it was a 6.7% contributor of the unknown fraction, 6.7 times 1,000 is 67 picograms. So we could go to 67 picograms and use this as a calibration curve, go up and say, if we had all 15 loci as we had in the original study, we would have expected about a quadrillion to 1. In this case, with these data, we only had 12 loci that were interpreted, and the loci are pretty random as to how much information you get. So dropping down 80%, we would predict about 1,000,000,000,000 to 1. Not the 10,000 to 1 that was being argued that all methods should have, but based on science and calibration, that we would expect about 1,000,000,000 to 1, and within random variation, we found about 100 billion. And so our findings were consistent with our own validation studies. When I talked about this in court, I basically quoted the literature on this and said that the less informative methods ignore some of the data, whereas if you use a computer to examine all the quantitative data, you expect a more informative result. And the best analogy here was saying, so suppose you make a microscope slide and you're trying to diagnose some cough. You want to determine if somebody has a cough and a fever, do they have pneumonia or not? So you make a microscope slide, you stain it, and now that's the data. How would you look at it? Well, if you look at it with the naked eye, you might see a slide. And if you looked at it with a magnifying glass, you might see lots of dots on the slide. But until you put it under a microscope, which is the appropriate resolving power for that particular data, you're not going to be seeing the various bacteria that are the causative agent. You could use an electron microscope maybe and see more. And so the point here is that each method has its own resolving power, and you want to use the appropriate measuring instrument for the appropriate data. This is what results look like. An inferred genotype at each of the loci is a probability distribution. These are screenshots from our true allele system. And you can also look at every locus and ask how much information was gained. On a logarithmic scale, there's 1, 10, 100. So you're seeing the rough gain of information, and it's logarithmic, so you add it up, and you get a number like 9 or 10. Or actually, what do you get? 11? 12? 11 or 12. And this is what data look like. So here's the victim's alleles, the 2 big peaks, and here are some smaller peaks. And the system, inside how it functions, tries to explain the data hundreds of thousands of times. It just keeps cycling through, trying out every possibility. And what you see is that if gray is the victim and blue is an unknown, that gives a pretty good fit to the quantitative data. Of course, the computer has to try out everything, so there are a lot of things with zero likelihood. But for Bayes' theorem, you have to be complete. And showing you in another interface that's good for reporting, you see if you look at the likelihood itself, at what turned out to be the matching answer, virtually all the likelihood is given to what the data, quantitative data, are indicating is most likely and some elsewhere. But the human review gives equal likelihood to many possibilities, many of which are completely impossible. And in that way, it dissipates its probability. And if you look at how it's computed, essentially, before you thought it was 3.5%, afterwards you thought it was more like 90%, that ratio is about 25, and the log is 1 point something, and you add them up. And we also showed that this isn't magic. Human review using the victim did about the same as the computer, and the computer did better in a few other cases. So the use of more information, whether it's the victim, which gave a lot of information to everyone, or quantitative data, which could account for stutter and other second-order effects, would raise up the information. Here you're looking at the different loci, say 13 loci. That's the likelihood of, or how much likelihood ratio of information is, and at every locus, how much information was gained. So what we learned from this case is that you can do an objective review that never saw the suspect, which generally would not be done by human review in a case like this. They would be making a comparison. It was easy to testify in court. We just explained what the computer is doing. It tries out everything, makes comparisons, and that which is more likely is given a higher probability. It was understandable. There hadn't been precedent before. Now there is, which is nice. And the whole goal is to preserve the match information in the data. The same way you expect the police to do a good job of transporting evidence and not lose 90% of it. You expect the lab to do good procedures and preserve the evidence. We also expect the interpretation to preserve the information that's present in the evidence. We were able to give an answer back to the attorney from, I guess, Friday afternoon to Monday morning, which isn't common in crime labs. Otherwise, there'd be no backlogs, right? And we found that presenting multiple match scores was helpful to the jury. It wasn't confusing at all because we could explain what the differences were, what the different assumptions are. And the prosecutor, Tony Krastik, felt that the DNA was really critical evidence in this, contributing to the conviction. I have a few general remarks. And I want to talk about why we do DNA in the first place and the implications of getting it right. For public safety, we want to have, we create DNA databases of criminal offenders. Have you all heard of ENDIS or CODIS, where millions of convicted criminals have their DNA stored in a database? Yeah. And then a police investigation can proceed by looking at hits against the DNA from evidence. And that can help prevent crime by catching criminals before they commit another criminal act. For example, with property crime, an active burglar will go about 10 crimes before they're picked up. Ray Wickenheiser wrote an interesting paper a few years ago, The Business Case for DNA, that showed that if you did proper use of DNA for all property crimes, sexual assault, and convicted offenders, you'd prevent 100,000 stranger rapes in the U.S. And no one has argued with his assumptions. If anything, with better methods, you prevent even more crime. So that's preventing crime is important to you. DNA can be a very big deal. Also, good DNA evidence can ensure conviction of the guilty, and also, as you find out in actual cases, avoid implicating the innocent when they're cleared with the DNA early on in the case. So I think there's an assumption on the part of the public, police, prosecutors, and so on, that the crime labs use methods that do everything possible to preserve all the DNA identification present in the data. You'd be surprised to find out that it was otherwise. But in fact, they don't, because, well, I'll go into because in a second. In fact, what happens is a lot of, if not most of the DNA identification information is discarded instead of being preserved. We already saw with these studies be millionfold loss of information. And what I want to show you is that with the gap paper, whereas the threshold comes down to 15 picograms, with the sort of review that was done in the case that I showed you, the curve shifts 1 log unit to the right, and the detection threshold increases from 15 picograms up to 150 picograms. So it's no surprise that the new guidelines that are coming down are discouraging labs from ever looking at completely analyzable crime-preventing DNA if it has more than about 150 picograms in it. The reason, because some of you are asking why wouldn't they do this, is there's really no incentive based on information, as you find when you speak with people in public policy at the National Institute of Justice and the FBI and other organizations. The NIJ funding for laboratories doesn't include information. We discussed that with them a few months ago, and they thought that would be a great idea for maybe 10 years from now. The accreditation standards don't have to do with the information you extract. The SWIG-DAM method guidelines, the new mixture methods came out 2 months ago, don't mention information. It's not part of the culture. It's just not what people do. Instead, the actual metrics and incentives have to do with eliminating backlogs. That's a worthy goal. Passing audits, which is very important if the lab wants to stay in business. And testifying in a comfortable way that doesn't jeopardize a case, but that doesn't promote science, that just keeps people comfortable. In particular, we've sometimes joked in the forensic community perhaps a pillow would do. But in particular, DNA identification information is not assessed. And if it's not assessed, as you know from evolutionary theory, it's not selected for, and there's no reason for it to ever be used as a criterion. A study done by John Butler, who's head of the DNA Mixture Committee for SWIG-DAM, done 5 years ago at NIST, National Institute of Standards and Technology, found tremendous variation looking at over 50 laboratories. The labs looked at the same data, and they gave scores ranging from tens of thousands, 4 log units, up to hundreds of trillions. Very few. Actually, only one lab, just the Massachusetts lab did that, which was 14 log units. Most were sort of toward the low end. But whenever you have the same data being analyzed by most of the labs in the US, with 10 orders of magnitude different information being reported, it suggests that there is not tremendous reproducibility in the methods. Okay. The result, and this is a paper that we're preparing with New York State, is a failure to identify. So far I've shown you what the results are for situations where the lab had a result. So we condition the other way. What happens when the computer has a result? Then what does the lab do? So we looked at 86 items of evidence, and here in blue, you see them ranked by information, log likelihood ratio. The average, the median, is about 10 to the 15th, or a quadrillion. It falls off below 1 million at the very end. So this whole sea of blue is the area. It's the total sum of information, log likelihood ratio, that the computer found. Now, on top of that, we have 3 different methods. Gray pretends it's not a mixture. Green uses the victim, and orange doesn't use the victim. And what we see, if you just ask where a bar even occurs, is that in over 70% of these items of evidence, no quantitative match score is even given to the evidence. It wouldn't be reported in many jurisdictions. It's ignored. No number can be given to it. And if you look at the area of the human review bars divided by the total area, you see that over 80% of the total information, the area of the log likelihood ratio, is thrown out by current human review practices. And you might think this is going to get better with newer guidelines, but in fact the result of 2 months ago is the thresholds are now being raised, and crime labs are now not allowed to report. So these numbers are only going to be getting worse. So what can you do about it? This is the last slide. So you can relax, loosen your tie, whatever. What's the scientist's task? What moral obligation do you have as a scientist? How many of you are thinking of going into science? That was a loaded question given the prior, the name of the symposium, right? So in your practice, if you practice science, like at a government laboratory, saying doing forensics, you should strive, I would think, to be doing the best identification science that you can, not practicing black arts of some kind, which came out of the NAS report on the state of forensic science in February this year. It's well recognized that there's a need for a more rigorous identification science. And you've seen how to do it here. You objectively infer all possibilities using computers and Bayes' theorem, and when you're done, compare them with a suspect. That's science. When you teach, as I've been finding out, there's also a burden to teach the public about scientific methods. This raging controversy in the scientific literature has been going on for 10 years about mixture interpretation. It's gotten very heated over the last year or two with many letters, angry letters, rude letters, being sent, I'm not involved, being sent back and forth from different practitioners across the globe. It's very vitriolic, and the press has no idea this is going on. The public's completely unaware, and police assume they just get all the information back from the evidence. But in fact, the top scientists know that there's a major crisis going on. So that public education isn't happening. I would advise you to learn more mathematics. The future is going to be in probability. Not to diss statistics, you still need to take statistics. But with computers, what computers can do with probability theory was not imagined by the Reverend Bayes in 1765. And what people have realized they can do, starting with the advent of the digital computer, has exceeded what anybody has imagined. In every field of science, social science, economics, which I guess is in between, statistical computing is now not just testing, it's basically not testing hypotheses true or false. It's giving accurate probability estimates in very complex problems with thousands of interacting variables. So you may want to start by taking maybe one probability class and see how it helps. And of course, in your research, it's not enough to say that there's a widely used methodology. It's helpful to assess its accuracy and its objectivity. There are many examples, but if the answer to a crime, here's the best example. I'll end with this. Suppose a friend of yours asks you what the speed of light is. Right? What's the speed of light? Anybody know offhand? Something like 186,000 miles per second. Suppose I told you, you asked me the speed of light, and I said, oh, it's around 2 miles per second. What would you think of that answer? Well, I can tell you, it's only off by 5 orders of magnitude. And most forensics answers are off by 6. So depending on what standard you're applying, if you're not interested in accuracy or objectivity, the speed of light might be a lot slower than you think it is. We have a lot of collaborators in our work from all over on these particular results. The main collaborators were from New York State with Dr. Barry Duquesne on the top right, Robin Cotton from Boston University, and Jake Adane from Carnegie Mellon University, and of course the team at Cybergenetics. Thank you. Questions? Sure? I can't hear, sorry.
Do you feel there are any ethical concerns with collecting DNA samples and storing DNA samples of criminal offenders and potentially citizens worldwide?
Those are lots of different questions. People have concerns with the DNA samples themselves. Sometimes people like to keep them to confirm or not. So if you're not keeping the samples, but you're only keeping the identification information, that eliminates a lot of the ethical concerns about will the government start some kind of genetic testing program. And so if you know that the material is disposed of, since the markers themselves are only from junk DNA markers that as far as we know have no or virtually no diagnostic value for genetics or insurance companies, that eliminates a host of problems. The question of what do you store on the database has been argued for at least 10 years. Some people, legal scholars, propose putting everybody on a DNA database because that would be fair. Other people, I think the state of Texas began with only people on death row, which didn't do much to prevent crime. But there's an interesting issue that gets raised with the notion of, and then in between you have major crime, minor crime, misdemeanors, arrestees. Many states have arrestees, not just people who have been convicted. That's causing ethical issues in Britain, which started the DNA databases over 10 years ago. But there's another issue that doesn't really come to the fore, and that is how informative, how accurate is CODIS, is the National DNA Database. Well, the way that CODIS has represented and will continue to represent information uses the weakest methods, just like the inclusion methods that I showed you. So the false positive rate currently is incredibly high. Now, if you use likelihood ratios, quantitative review, and so on, you'll typically either get the one correct match or nothing. Currently, police will routinely, on a low stringency match, come back with hundreds of names, of whom none or one might be possible. And that's with a small database of only convicted offenders of maybe 10 million. So if you scale that up to the whole country, the real ethical concern is the information failure on the part of the DNA databases and the DNA interpretation methods, which would essentially make every American citizen a suspect, probably within 1 year, for some major crime in the country. So at that point, it becomes useless, I guess, to one sort of regime, or maybe useful to another sort of regime. It depends on the regime. But if you want to live in a country where you want to have everybody in a database and you don't want everybody to be a suspect, you sort of have to start all over again and use more informative probability methods based on science, not luck, So that's the ethical concern there, is how good was the science. I don't think the science was good enough to go to an all-citizen database. Not the answer you expected, right? Any other questions? Thank you.